The overall goal of this protocol is to introduce X-ray crystallography methodologies applied to microcrystals, and particularly to those produced in cells, which are considered in vivo crystals.
The main advantage of this approach is that it provides a simple and efficient process to produce, detect, and analyze microcrystals. This method aims at facilitating the use of microcrystals by using crystals grown inside living cells, which removes the need for complex purification process and empirical crystallization trials.
In this video, we will be focusing on sample preparation and manipulation for X-ray diffraction experiments at a synchrotron beamline, which are the steps that differ the most from classical crystallography.
In this demonstration, we will be using crystals from the viral polyhedrin protein grown in insect cells, but the method can also be applied to proteins forming crystals in any bacterial, insect, mammalian, or yeast cells.
First, incubate 30 milliliters of SF9 cells overexpressing the cypovirus polyhedrin protein for 3 days at 27 degrees Celsius on a rotating platform. Transfer 500 microliters of SF9 cell culture into a microcentrifuge tube using a sterile serological pipette, taking care to ensure that the sterility of the main culture is maintained by using a Class II biosafety cabinet and following standard aseptic techniques. Pipette 5 microliters of cells from the microcentrifuge tube onto a glass slide. Then, gently angle a glass cover slip over the sample with one edge touching the drop, and carefully lower the cover slip onto the liquid, avoiding the formation of air bubbles.